Now let's discuss the second class of enzymes called oxidizing enzymes. As the name suggests, they are involved in the oxidation reactions, and by oxidation mean it is meant that loss of electrons take place from a compound that is known as oxidation. We can observe this in the case of burning candle. You can see the wick inside the candle. When it is burned, it is converted into water and carbon dioxide. So you see it disappears. So this is an example of a oxidation reaction, that the products get converted into simpler forms, or rather much simpler forms, as byproducts. So oxidizing enzymes, they can bring a lot of deterioration or changes in the food. We can take examples. For example, in plants there are certain oxidation enzymes such as peroxidases, ascorbic acid oxidases, tyrosinase, and polyphenolases. These are the ones which are responsible for undesirable reaction in plant tissues or fruit tissues or vegetables. We can look at them one by one. These peroxidases normally acts upon phenol compounds in vegetables. One of the example is horseradish. This is a vegetable. If it's cut down, the surface gets discolored after some time, and it becomes darker in color. This is because of the action of these oxidizing enzymes called peroxidases that would be breaking down the phenolic compounds and making them into dark color end products. So that would bring up an undesirable change that can be seen in many other vegetable surfaces as well. Ascorbic acid oxidase, as you know that ascorbic acid is commonly known as vitamin C. It is there in fruits and vegetables. If this enzyme acts upon ascorbic acid, that would mean that it would break down ascorbic acid into simpler compounds. So we would be losing the vitamin C activity in fruits if we have these enzymes active in the product. Similarly, peroxidases are another group of compound. They do not directly act upon vitamin C, but their byproducts, when they form different byproducts, these byproducts might attach with vitamin C and degrade vitamin C into other products, simpler products. So this is how both these enzymes are actually affecting the nutritional value of food by reducing the vitamin C contents. Phenolases, as the name suggests, that it acts upon phenolic compounds within plant tissue such as fruits and vegetables, and these phenolic compounds, once degraded, they would be producing darker color tissues, and these dark color, as you can see in many fruits, when they are cut and exposed to oxygen for some time, they would change their color, and the color might be quite undesirable because it would suggest that the fruit or vegetable surface is not fresh and it has been stayed there for certain amount of time, and it's not safe to eat such products. Although the safety are not a serious concern on action of these enzymes, but it's the physical or visual appeal that is affected. Another enzymes are tyrosinase oxidases. They work on tyrosine. As you know, tyrosine is an amino acid, and it produces dark color compounds. These compounds, once they are acted upon by tyronases, they then polymerize together, and these compounds, the end product, when polymerized, they make up another products called as melanin. These melanins or these colored compounds, when they are concentrated, they would give undesirable color changes within the end products. So this is how it would be actually affecting the quality of these product or shelf life of these product. This tyrosinase acts upon, it causes, especially it works on cut surfaces, and is same is true for many other enzyme that we already discussed. They would be acting when the surfaces are cut, because once we cut the surfaces, it would lead to the bleeding or the active material coming out from the cellular structure or the tissue structure. These compounds are released, and then they act along with these substrate. So the enzyme substrate comes together, it works on them, and the deteriorative process are speeded up. So this enzyme, one of the application of this enzyme is to know that it is present in shrimps, and in shrimps normally they would be producing black spots, which are undesirable reactions that take place. It happens especially at the tail region when they are especially at the junction of two overlapping regions. So we can see those black spots or black lines. They are there because of the enzyme called as tyrosinase. Tyrosinase is also present in calms as well. This is more profound, as I said that in the previous example, but calms would have also these tyrosinase activity. But if we have these calms, if they have been not treated well after catching from the sea, if we don't heat them, these tyrosinase would act upon these products and they would be discolored, and the product it would not have desirable quality. So that's why they must be deshelled or shucked immediately after they are harvested, and they must be heat treated to inactivate these enzymes that would increase their quality, desirable color, and the shelf life. These reactions are again not the normal metabolic functions. It only happens when these shrimps or lobsters or clams, when they have been their surfaces are cut. So that is the time when the enzyme substrate comes in together, and they start working and they start doing these undesirable reactions. So this was a little about oxidizing enzymes and their application.